Immunology, cellular components. So innate and adaptive immune. So in an innate immune, the components are neutrophils, macrophages, monocytes, dendritic cells, natural killers, lymphoid organs. The natural killers are lymphoid organs. We have the complement and physical epithelial barriers like the brain barrier and secreted enzymes. These are the all components. So we got neutrophils, macrophages, monocytes, dendritic cells, natural killers, complement, and physical barrier and enzymes. So the mechanism of action is germline encoded. Germline encoded. So you was born with that. Germline encoded. So germline encoded refers to the genetic information passed down through the germline, the line to line. It means the line is productive cells that produce the sperm and eggs. So you inherit that. This means that the genetic information is inherited from parents and present in all cells of spring. So germline encoded. So the response to pathogens is non-specific. Occur rapidly in minutes to hours, and there's no memory response. That means if you have been exposed before to the same bacteria through this innate immunity, you won't be like remember like, oh, this bacteria, we get infected last month with this bacteria, let's fight faster or harder. They don't remember. So this innate immunity compared to adaptive immunity, this one doesn't have memory response. The other one has developed memory response. We're gonna see that part in a minute. So secreted proteins are lysozymes, complement, C-reactive protein, defendins, and cytokines. Remember, the cytokines are proteins that signal cells to communicate and regulate the body's immune system. They are released by many different types of cells, including immune cells, thromboblasts, and endothelial cells. So remember, we have cytokines like pro-inflammatory cytokines, and we have anti-inflammatory cytokines. The very examples that we have is interleukins S, chemokines, interferon, tumor necrosis factor. All those kind are different kind of cytokines. Okay, those are the cytokine. Remember, lysozymes are organelles with cells that their function is a self-digestive system. Those organelles contain enzymes to break down large molecules and recycle their components. In some cases, defect in lysosomal function can lead to lysosomal storage disease. There's some kind of group of disease that come up from lysosomal storage disease. So lysosomes functions are essential for cell digestion. They break down various materials, including material taken up from outside of the cell through endocytosis. Remember, endocytosis bringing things inside the cell. The cellular waste product and damaged component through autophagy. So they lyse that. So their name said lysozyme. That's what they mean. Lysol. They lyse. They break materials. So they break materials. They break waste product. And then they have a component in pathogen disease. So they do digestion. They do digestion. Some kind of pathogen. We're gonna see that later in the textbooks. But the most important that USMLE always asks us is the lysosomal storage disease. So there's a group of lysosomal storage disease. We're gonna see it later. We got like a Gaucher disease, Fabry disease, Pompe disease, mucopolysaccharidosis disease. Those are all group of lysosomal disease when there's like this lysosomal storage disease. So remember, the lysosomal storage disease is caused by defect in the lysosomal enzymes or related proteins, leading to the accumulation of undegraded sustains with the lysosome. So the lysosomes cannot destroy all the material, and they get storage inside the lysosomes that give you all that kind of disease: Gaucher disease, Fabry disease, Pompeii disease, and mucopolysaccharidosis. It's hard to say. Mucopolysaccharidosis. So another is the component. We have the component here too, the C-reactive protein. In the other side, the adaptive immune system only have immunoglobulins and cytokines that they're in both. So what happened in innate immunity? You was born with all this process here. The key features in pathogen recognition is the toll-like receptors. There's a pattern recognition receptor that recognize pathogen and damage associated molecular pattern. Pathogen associated molecular pattern and damage associated molecular pattern. Those two do activation of NF-κB. This is a nuclear factor kappa B. So there's going to be an activation of nuclear factor kappa B. This one release pro-inflammatory cytokines. By example, so they can recognize in some bacteria or virus some LPS, like a superficial antigens that they recognize, and they activate the whole process of destruction. Compared to the other side, in the adaptive immune, instead of happening this whole process, that in this case what happened is it's going to be memory cells. Those memory cells recognize the pathogens before, and they act against. So in the adaptive immunity, we have the T cells, the B cells, and circulating antibodies. B cells and T cells. So the mechanism of action is there's going to be variation through BDJ recombination during lymphocyte development. So the BDJ mean variable diversity joining rearrangement. It is the variable diversity joining rearrangement. So this occur in the early stage of T and B cells maturation. It is going to be like a in the lymphocytes, or only in lymphocytes B and T. This recombination occur primarily in lymphoid organs like at bone marrow, B cells, and thymus. This is some kind of like a rearrangement in the mean. So you're gonna have a lot of variety of immunoglobulin. So it's like a spread and different kind of immunoglobulin. We're gonna see that in the book later. We're gonna read it later. But this is like a they explain it like a rearrangement, variable diversity joining rearrangement, recombination. So you don't get only the one that you get when you was born. So in this case, you can have like a spread and different kind of recombination to attack different kind of bacteria. And if you recognize those bacteria before you do that kind of recombination, so you can have a memory, and when you recognize that bacteria again, you can attack it again. So response to pathogens, the way they respond to pathogens is highly specific. This adaptive immune component on the immunity is highly specific. It's refined over time. So the more you get exposed to different bacteria, different virus, it get refined. It get more production of interleukin, immunoglobulin, the different kinds. So if you get affected with that virus again or that bacteria again, you can have memory, and they're gonna have a response faster and more robust, more stronger. So it's highly specific, refined over time, develop over long periods. So you got immunity for a long period, let's say years or month to years, and you're gonna have memory response is faster and more robust. Okay, so secreted proteins in this case, remember cytokines are in both, and immunoglobulin. So the most you have in this one is immunoglobulin. So remember, every time they talk to you about immunoglobulin, they're talking about adaptive immunity. Adaptive immunity. So you wasn't born with immunoglobulins. So you had to create a vast number of immunoglobulin. So that's why they create this recombination. We're gonna explain it later how this recombination is gonna happen. But immunoglobulin, remember, we have immunoglobulin G, we have immunoglobulin M, all that kind of immunoglobulin. Okay, so we're gonna read this later. So the key features for pathogen recognition is the memory cells, activated B and T cells, subsequent exposed to a previously encountered antigen, give you a stronger and quicker immune response. So remember, if you are exposed to the same bacteria again, let's say 2 or 3 months later, you're gonna have a more immune response, stronger. This is the same case when you have like an allergic reaction. Let's say you touch some kind of you touch some kind of plant in the wood, and you won't get any reaction. But 2 or 3 months later, you touch the same leaf, the same plant, and you're gonna have a big reaction. This is they explain that's why, because you have memory cells, activated B and T cells, subsequent exposed to a previously encountered antigen, and they're gonna give you a stronger and quicker immune response. That's sometimes why you explain some people that they say, I have eaten before fish, but I wasn't allergic, but now I'm allergic and I don't understand. So this is some kind of explanation. So the adaptive immune response decrease with age. So the adaptive immune response decrease with the age. This is called immunosenescence. Immunosenescence. So this is the key factors different from innate immunity and adaptive immunity. Remember, adaptive immunity is more stronger and faster. When you second expose, because this one say here that is more rapidly. This is more rapidly. But what I mean faster here, faster when you have been exposed before, because it's a memory response faster and more robust. Remember, in this case, the protagonist of this one is immunoglobulins, and the different kinds of immunoglobulin is through the process called VDJ, variable diversity joining rearrangement. Variable diversity joining rearrangement is a recombination of all this kind of immunoglobulins. So if you don't have this process and the right process, if you don't have this process right, you won't have immunoglobulins, and you're gonna have a whole bunch of disease that we're gonna read later. So here's they talk about immune privilege. Immune privilege refers to the organs and tissues where chemicals and physical mechanisms limits immune response to foreign antigen to avoid damage that would occur from inflammation sequela. Allograft rejection at those sites is less likely. So this is a group of organs more likely that has immune privilege. So we have the eye, brain, placenta, and testicles. We have the eye, brain, placenta, and testicle. a good way to remember is through my brain, no, through my eyes I saw a girl, and my brain get in love with her. We played with our testicles, and then we got a product in our placenta. I saw a girl with my eyes, my brain get in love with her. We play with our testicles, and now she has some product in her placenta. I don't know, that's a good way to remember, but it's not difficult. It's eyes, brain, placenta, and testicles. Those are tissues in immune, they have immune privilege. So the immune privilege mean because they have like a chemical reactions or physical mechanism. For example, the brain have the brain barrier, so they have a physical mechanism. So the immune system is not that strong in those organs. For example, the eyes have very tiny organs, very tiny. They need very tiny parts that if you have an inflammation inside the eye, you're gonna have losing your vision. So they have to be like a mechanism so the immune system doesn't be that strong in those organs. Let me read what is immune privilege. So immune privilege refers to a phenomenon where certain tissues in the body, like the brain, eyes, are less susceptible to immune response compared to other tissues. So this privilege allows those tissues to maintain homeostasis and function without excessive immune activation, which could be detriment or damage to their structures. Let's say, example, the eyes. If you have like inflammation in your eyes, you're gonna lose your vision. So for any inflammation that you have in your body, you're gonna be losing your vision. So that's why the eye is one of those organs. So that's why in this case, the eyes, they can have like a tolerance to allograft when they have like transplant for another person. The cornea is very likely that you won't have any rejection. So in some privileged tissues, like example the eyes, allograft transplant tissues from another individual can be accepted without any need for extensive immunosuppression. This is seen in corneal allograft on transplant. They're doing a lot of investigation about this process of immune privilege because they couldn't understand. If they could understand, the immune privilege could lead to a new strategy to treatment immune disease. So remember, the eye have immune privilege to protect the vision by limiting destructive inflammation. The eyes, like the brain, is this immune privilege is achieved through the combination of anatomical barriers and immunosuppressive molecules and regulatory mechanisms. Another thing that could be important on your test, they could ask about immune privilege. It is or like a which of the next mechanisms explain why the eye have an immune privilege. So the lack of efferent lymphatics, the lack of efferent lymphatics, and the presence of a tight blood-retinal barrier restrict the immune cells entering. So anatomical barrier, immunosuppressive molecules, and regulatory T cells are the three more mechanisms. That's why the eye has a immune privilege. So I don't know. I could find this interesting for the test. So do you remember? You remember the test? They always give you some questions that you never seen in the first aid on using the word you never seen. So that could be from Google. This is important. I just put it here just in case you want to read it. So the immune privilege refer to the ability to certain tissues and organs to tolerate the presence of a foreign antigen without eliciting a typical immune response. Those projections then from the potentially damaged inflammatory process. So the key aspect of immune privilege for the USMLE, the purpose is to prevent damage to vital tissues and organs, for example, the brain, eyes, and testicles, by limiting the inflammatory response to the foreign antigens. So the mechanisms, the physical barrier we just said before, you can read all of this, immunosuppressive environment, limited antigen presenting. So example of immunoprivileged sites. So we have the brain, protected by the blood barrier and other mechanism. The eyes, particularly the retina, has high level immunoprivilege. Important, the retina has the high level immunoprivilege. It's important. I think that's a good question. The testicle protects foreign tissues graft from rejection. Remember, when you have a testicle cancer, never do a biopsy. You have to do a. This is very important, and I have seen a lot of questions. In general, testicular biopsy is not recommended for diagnosis of testicular cancer. In case if you have a mass, testicle mass, they recommend to do osteotomy. They don't recommend a biopsy. Just keep that in mind. The most important is why you can have the risk of spreading the cancer. Taking a biopsy, essentially through the scrotum, can potentially spread cancer cells and complicate future treatment. Remember, when you do a biopsy from the testicles and you bring the biopsy through the scrotum, remember the scrotum have a different lymph node. So the scrotum, remember the lymph node that we checked before is the superficial inguinal. Remember the superficial inguinal is that they drain from the scrotum. So you have a big chance to spread the cancer in this case. Keep that in mind. And as we said before, eyes, testicles, and placenta. Remember the placenta is essential for the fetal development and survival. So now we're gonna read about Mayer histocompatibility complex 1 and 2. Mayer histocompatibility complex 1 and 2. This is a little complex to understand for a lot of people. We're gonna be explaining them most we can. Let's see. So the Mayer histocompatibility complex 1 on 2. So the Mayer histocompatibility complex is a group of genes that encode sulfase proteins, crucial for the immune system ability to recognize self and non-self. So the proteins known as the Mayer histocompatibility complex molecules are involved in the antigen presenting, where they are displayed peptide fragment to T cells. The human version of the Mayer histocompatibility complex is called human leukocytic antigen, HLA complex. Human leukocytic antigen, or HLA complex. So the antigen presenting is a crucial process in the immune system where antigen presenting cells display fragment of antigen, like protein from pathogens or their surface, or allowing T cells to recognize and initiate an immune response. So the Mayer histocompatibility complex molecules are cell surface protein, essential for the active immune system. Acting as antigen-presenting molecules, they play a role in recognizing and distinguishing between self and non-self antigen, allowing the immune system to respond effectively to foreign invaders like pathogen or tumor cells. So they are two groups. So the Mayer histocompatibility complex encode for HLA genes. So they present antigen fragment to T cells and bind T cell receptors. So we have two groups, the Mayer histocompatibility complex 1 and Mayer histocompatibility complex 2. We're gonna call MHC1, MHC2. So the number 1, it's localized or it's expressed in all nucleated cells. Its expression is all nucleated cells. Antigen presenting complex or antigen presenting cells, platelets, and just exception is the red blood cells, so they are not presented in the red blood cells. This means antigen presenting cells. So they are different groups. They're the loci is the HLA-A, HLA-B, HLA-C, MHC1. Loci have one letter, so remember this. So they have one letter, A, B, and C. So to remember, the step 1 tell you, the first 8 tell you that one loci have one letter. So HLA-A, HLA-B, and HLA-C. Remember, HLA is a human leukocyte antigen. Human leukocyte antigen A, human leukocyte antigen B, human leukocyte antigen C. The other group have two letters: human leukocyte antigen GP, human leukocyte antigen GQ, human leukocyte antigen DR. Remember, MHC loci have two letters. So that's the main difference, so you can recognize each one. The banding is the MHC1. It's bind to TCR and CD8. Just remember, the one is bind to the CD8. That's why they do this mnemotechnic here, telling you that CD8 bind to MHC1 is equal to 8. The same thing here, the equation, because this one is united to CD4. So CD4 plus MHC2 2 is number 8. So this is some kind of mnemonic, so you can remember. But just remember, TCR and CD8 are a unit to MHC1, and TCR and CD8 they bind to MHC2. So remember, this side is for cytotoxic T cells, and the other side is for helper T cell, and cytotoxic T cell. So the structure is one chain, one long chain, and one short chain. So you can see here one long chain and one short. And the other side, there's two equal long chains. So one long chain here, and the other side another long chain. So there you have two long chains. You have 2 alpha and 2 beta. In the case of MHC1, we have one long chain for one short chain, and remember, we have inside 3 alpha and 1 beta. So 3 alpha, 1 alpha here, second alpha here, and 1 beta, this one here. So they have 3 alpha, 1 alpha, second alpha, and third alpha here. And then they have 1 beta. Just recognize that this is the MHC1. So in the other structure, MHC2, so we have 2 alpha and 2 beta. So 1 alpha here, another alpha here, 2 alpha, and then 1 beta here and another beta here. So 2 alpha and 2 beta. Remember, MHC2 have 2 alpha and 2 beta in their structure. Okay, so in their expression, they express in all nucleated cells. This one, MHC1, is expressed in all nucleated cell, and APCs, antigen presenting cells, and the platelets, except in red blood cells. And the other side is present in all APCs. Okay, so the function is, as I said before, the function is they present endogenous antigen. Endogenous. This one is endogenous. The other one is exogenous. Exogenous antigens. So the endogenous antigens, like a virus or cytotoxic protein, cytosolic protein, viral or cytosolic protein, they bind to CD8 plus cytotoxic T cells. So CD8 is the same one called cytotoxic T cells. So in the other side, in MHC2, the function is they present exogenous antigen, exogenous antigen, exogenous. Remember, exogenous. And the other side is endogenous. So makes sense if you think endogenous, because the structures is more a little tiny. They have to be more tiny in order to find those those antigen endogenous. So the other one is more bigger. So she can walk around. She got 2 legs. She can walk around finding those exogenous antigens. Okay. So this one number 2 is exogenous. It's extracellular, present exogenous antigen, and you can see the structures is bigger and have 2 legs, so she can walk around trying to find those exogenous antigens. So the other one only have 1 leg, can be walking around. It's going to be inside the cell. That's why it's going to present endogenous antigens like a virals, cytosolic proteins, and they bind to CD8. Remember, CD8. The other one bind to CD4. So the antigen loading. Remember, they are both in the extracellular surface. They're both in the extracellular surface, but one present endogenous antigen, the other one present exogenous antigens. So one way to remember doesn't mean that one is intracellular and the other one is extracellular, but they present different kind of antigens, endogenous and exogenous. So one way to remember is that this one have 2 legs, so it present exogenous antigen. But one way to remember is like she can walk around trying to find exogenous antigen, but in reality she doesn't walk around. She just stays in the extra surface space. But it's a good way to remember because I have two legs, saying that it walk around. Okay, so the other one only have 1 leg. This one doesn't walk around. You have to stay there attached to the extracellular space or surface in the cell membrane. This is a good way to remember that she only can present endogenous antigen. She can be walking around trying to find external antigen. So this is a good way to remember. So another thing is that it's associated with a microglobulin. This microglobulin, you can see B2, it's gonna act like for you to remember, like it got a cane, a little cane here to help her. So it only have 1 leg, but then there's a little molecule here that help her to stand. It looked like a little cane. It helped. This is going to be B2 microglobulin. Okay, is a associated protein microglobulin. You can see like a little cane. So this one is in MHC1. Remember, MHC1 only have 1 leg. MHC2 have 2 legs. Okay. So in antigen loading, this is very tiny explanation here. There's a more complex explanation now what happened really here. You can see in a video. I'm gonna put it on the camera right now. I'm gonna put it here so you can see a better explanation, more extended, what really happened here in antigen loading and presenting. So what first aid said is antigen loading, antigen peptidos. The antigen peptidos loaded into the MHC1 into the endoplasmic reticulum. after delivery via TAP, transport associated with antigen processing. So you're gonna I'm gonna show you the video so you can go and watch it, because it's more explained. So just remember, from MHC1, the TAP is the more important molecule that give you the transport into the reticulum and the plasmatic reticulum. And in the other one, in MHC2, the most important molecule is the invariant change. The invariant change is the molecule that bring the antigen into the endosome. Let me show you the video so you can go to check. So this is a video I told you about. He explained very well the whole process. I made is the compatibility one, specifically the one. So you can go, his channels is Armando Hasudungen. He got like a 2 million subscriber, and he just type his name, immunoglobulin MHC1 processing. So you can go to check definitely.
Cellular fluid, and here we have the intracellular fluid. Some important components within the cell.
I cannot put the whole video because this is
endoplasmic reticulum.
they don't want us to
endoplasmic reticulum. And of course, the rough endoplasmic reticulum is close to the nucleus, which I have not drawn. But just know that we have an endoplasmic reticulum. Further, we also have the Golgi apparatus, which is important in the MHC1 processing.
Definitely go to check that video. It's very well. It gave you very graphic and very well informed. So thank you. So next, what we're gonna read is a human leukocyte antigen subtypes associated with disease. So I remember in my old first aid, it used to be like A3, other one, DR5, but I don't know what they put on another place of the book, but they're not here anymore. Just remember the A3 is associated with the hemochromatosis. We have B27, HLA subtype B27 is associated with psoriatic arthritis, ankylosing spondylitis, AVD associated with arthritis, and reactive arthritis. So it's important to remember mnemonic is pair, a pair. B27 pair. So another one is DR3. DR3 is we have here diabetic mellitus type 1, lupus erythematosus systemic or systemic lupus erythematosus. We have to Graves' disease, Hashimoto thyroiditis, Hashimoto thyroiditis, and Addison disease. Remember this one. There are three, DR3, diabetic mellitus type 2, systemic lupus, Graves' disease, Hashimoto thyroiditis, and Addison disease. Diabetic mellitus type 1, remember, HLA 3 and 4. 1 plus 3 is 4, as SL3, some kind of mnemonic. And there are DR4 is important for rheumatoid arthritis, diabetic mellitus 1, and Addison disease. The good mnemonic is the room have 4 walls. There are 4 walls in one room. So DR4 rheumatoid arthritis is gonna remind you that there are 4 walls in one room. So there are 4. Remind you that there are 4 on room rheumatitis, and there's in 4 walls in one room. Remind you the diabetic mellitus type 1. There's another very interesting video about this. For remember all of them is let me find it here. So this video from Dirty Medicine. He used to call before Dirty USMLE, but now he changed his channel's name to All Dirty medicine. This is a video from 5 years ago, but it's very high yield, very high yield. This video explained a lot about A3, B27, and DQ and DQ8, DQ2 and DQ8. There are no in the new book, but they are in the old books. So it's important to know anyway. So watch this video. This video really interesting and explained very well. Explain it very well, so you can watch it.
And it was in the gray color. It was in the iron color in Dirty USMLE's video. So it's hemochromatosis. Bam. Free point. Love you guys. Let's keep going. HLA-B8 is associated with myasthenia gravis, Addison's disease, and Graves' disease. So I want
Interesting
you to think
video.
about that.
Very interesting video. So I highly recommend to go to watch his video. Because explained very well, at least the mnemotechnic to remember all of this. DR4, DR3, B27, A3, DQ2, DQ8. They're not in this book, but they are in the old book. Let me see if it's a little lower. They probably moved to another part of the book, but it's very interesting, that video. So I wish you go to check that video. So let's see if I can do a couple questions before jumping into the next video. So in the next video, we're going to be reading about this part here: measure function of natural killer cells, measure function of B and T cells, and differentiation of T cells. Okay, let's do a couple questions. So we have here a question that is related to something that we just learned today. So here said, 5-year-old girl is brought to the office for evaluation for a persistent cough. The patient have a history of productive cough daily for past month. History include a recurrent episode of otitis media despite bilateral ear tube replacement, numerous lower respiratory tract infections, and occasionally ulcerative skin lesion. So laboratory evaluation showed normal levels of total B and T cells and serum immunoglobulin. So genetic testing revealed a mutation in the TAP1 gene, which encodes a protein involved in the transport of cytosolic molecules into the endoplasmatic reticulum. It sounds familiar. Which of the following process is most likely to be impaired by this mutation? So which of the following process is most likely to be impaired by this mutation? So B cell differentiation, cytotoxic T cell activation by MHC class 1 molecules, destruction of phagocytosis organelles, MHC class 2 molecular expression on B cells, and migration and extravasation of neutrophils. So in this case, we pick which one? So remember, the TAP1 is the transport that facilitates the transport. So in this case is B, cytotoxic T cell activation by MHC class 1 molecules. Perfect. Okay, so that's the answer. So let's see the explanation. So we can see some graphic here. You can see class MHC pathway, CD8. You can see the TAP proteins are the one who bring the molecular from outside and bring it into the extracellular pathogen. You can see go through the proteasome and leaving in a fragment, more small fragment, and then with the help of the TAP protein get into the reticulum endoplasmatic. Then inside, this is just kind of like the video we just recommend to the guy to check. He explained everything very well, this part. This is another good view, better view. So transport associated with antigen processing, TAP protein are transmembrane protein necessary for the presentation of cytosolic antigen on major histocompatibility complex molecules. When cellular proteins or pathogen derived from protein are degraded to proteasomes, some of the result peptidose fragments are transported into the endoplasmatic reticulum by the TAP proteins. unloaded into the MHC class 1 molecules. The MHC class 1 peptide complex then translocate to the cell surface where they can activate cytotoxic T cells through the interaction with T cell receptor and CD8 co-receptors. So correlated with this case, this little kid, so the activated T cells clonally expand and kill damaged or infected cells by release cytokines that induce apoptosis. So genetic mutation of the TAP1 affecting the MHC class 1 peptide complex expression are rare but can cause ulcerative and granulomatous skin lesions and frequent respiratory infection. That's the case of the question. Normally, normal lymphocyte immunoglobulin levels. So remember, everything you have like those kind of infection on granulomatous skin lesions and ulcerative lesions, and the like leukocytes and T cells and B cells are normal, immunoglobulins are normal. So you have to think in that TAP mutation, gene mutation, genetic mutation of TAP. Thank you. See you in the next video.